Warm wishes and welcome to the session. So today myself Husna Khanam and I am from Department of Biochemistry, Justice Academy of Higher Education and Research, Mysore. So today in this session I'm going to explain you all about the nitrogenous bases. So let's understand the basic concept what exactly the nitrogenous bases are. Nitrogenous bases is nothing but, or a nitrogen base is a molecule that contains nitrogen and has the chemical property of a base. A base is nothing but it's a substance or a molecule that can donate a pair of electron to other element or a molecule and it form a new molecule. In this way, nitrogen bonds with carbon, hydrogen and oxygen atoms. These different elements can arrange themselves in a ring and we name the compound based on its ring formation. So, nitrogen bases are also sometimes referred as nucleobases. The important bases, especially it's very important in biology because they are the key components of nucleotides. Nucleotides are the building blocks of genetic information like DNA and the RNA. So coming to what exactly how it's very helpful in our human system or in a living system is that our genetic information holds all the instructions and all the different kinds of functions of our cells and the functioning of our body. Changes in which or changes in the nitrogen base will cause a disastrous consequence in a human being or in the living system. It causes mutation. Mutation is non-curable, non-resistable change. It leads to cancer, sickle cell anemia and numerous different types of genetic disorders. So nitrogen bases come in two forms, that's purines and the pyrimidines. Purines are nitrogen bases which have two ring structure fused together and the pyrimidines are the single ring structure. Purines are as you can see in the slide where I have given a general view of a purine and a pyrimidine, where purine have the two ring structure bound together and the pyrimidine have a single ring structure. So moving forward, basically purines are much more larger than pyrimidine because of double ring structures. Basically purines and pyrimidines serve as a basis for the building blocks of DNA and RNA. In purines, there are two kinds, that's adenine and the guanine, and in pyrimidines, it's cytosine, thymine, and uracil. So let's understand it a bit deeper and starting with adenine. Adenine is a nitrogen base represented by a symbol A in the DNA double helix model or an RNA model. So here guanine is a purine structure where two different kind of rings are fused together to form a purine or a molecule. This adenine attaches to the sugar molecule in nucleotide through the first carbon on either ribose or the deoxyribose sugar. It has two binding sites that forms a complementary bond pairing with by hydrogen bond with other nucleotides. Basically adenine always pairs with thymine and coming to guanine, it's represented by G and it is also a purine ring structure where two rings are fused together to form a molecule. Guanine is way too similar to adenine however it contains a carbonyl group instead of an amine group and an amine group instead of hydrogen atom as a substituent in the larger ring. Guanine has three binding sites which allow it to form hydrogen bond with its complementary nucleotide. So coming to thymine, it is a pyrimidine ring structure, that it contains only a single ring structure and it's denoted by the T in the models of DNA and the RNA. Unlike adenine and the adenine guanine, thymine is a pyrimidine model and thus it has only one ring structure. Thymine is sometimes also known as 5-methyl uracil because it has a methyl group substituted for the hydrogen atom of uracil on the 5th carbon in the ring. It has 2 binding sites which allow it to form a complementary base pairing with the help of hydrogen with other nucleotides. And comes to cytosine. Cytosine is also a pyrimidine molecule where it contains only a single ring structure. It is denoted by the structure C. It contains in both DNA and the RNA. It will be having 3 binding sites to form a hydrogen bond and its complementary nucleotide to bind with the complementary nucleotide. Coming to uracil, it's kind of advanced structure where it is only present in the RNA in place of thymine. Thymine is not present in the RNA, so in place of thymine, it's present in RNA. So adenine always pairs with uracil in when comes to RNA, and guanine always pairs with cytosine, and it's vice versa. So it contains possibly 2 binding sites that allow it to form hydrogen bonds with its complementary bases, as you can see in the figure where the RNA strand showing the complementary base pairing, and in the right and in the left hand side there is a structure of uracil where you can see the single ring structure containing different types of functional groups. So let's understand the nitrogenous base pairing. Nitrogenous bases are always complementary. It connects in group of 2, as these nitrogen base pairs is the way because DNA is double stranded and the pairing allow for the safe transfer of genetic information or the information from DNA replication transcription until the process of transcription, the process that makes RNA. Transcription is nothing but the process that makes RNA. So the specific order of bases in DNA is what transmits the genetic information. The genetic information is copied from DNA to mRNA through the process of transcription, where base pairs are matched with their complementary RNA nucleotide by enzyme such as RNA polymerase. The mRNA is then edited and transferred out of the nucleus to the cytoplasm. That translation occurs. In the cytoplasm, the RNA is then read by the ribosomes in the cell and groups of 3 bases called the codons. Each codon consists of 3 nucleotides. Thus, this forms 3 bases. Each codon codes for a specific amino acid. Amino acids are the building blocks of proteins. The transfer RNA, or it's a kind of RNA. It's a kind of RNA that have anticodon sequences that allow it to bring the correct amino acid sequence to the ribosome. Let me explain you what exactly is the anticodon sequence. See, for example, in an RNA molecule, if a strand is a codon, then the strand pairing with it will be anticodon. Like if in the codon strand the bases are adenine and cytosine, then in the anticodon strand it will be thymine and the guanine. Likewise in the vice versa, like it should bond with each other, so it's an anticodon. The opposite kind of nucleotide will be present in the anticodon structure. This is very important in the formation of proteins. The ribosome then connects the amino acid using peptide bonds to form a polypeptide chain. When a stop codon is reached in the mRNA, the ribosome releases the polypeptide chain. The stop codons are also called as termination codons. These are the kind of special codons which gives a signal to the ribosome. It gives a signal in the ribosome to the mRNA that the need of protein is sufficient. It can stop the processing of or the processing of protein. So at that instant, the mRNA releases the polypeptide chain with the help of additional chaperone proteins. Chaperone proteins are nothing but these are the kind of proteins that folds into a three-dimensional shape, and it allowed to do its job in the cell. Thus, the order of bases holds the information of both the structure and the function and the capability and all the different kinds of a process of transcription and translation. So moving forward, let's understand how the linking of DNA and RNA with base pairs takes place. As I explained earlier, purines and the pyrimidines are the basics of building basis and the building block. These are the basis for the building block of DNA and the RNA. DNA is double stranded and the RNA is single stranded structure. So basically, it is a molecule that's responsible for coding, decoding. Basically, DNA is responsible for life, instructions of life, whereas RNA is responsible for coding, decoding, regulation, exploitation of genes, etc. etc. These instructions are encoded in order of nitrogenous bases all along DNA and RNA molecules. These bases are starting from either the single ring pyrimidine or in the double ring purine. Then some extra nitrogen, hydrogen or oxygen atoms are added on the basic ring to form a nitrogen bases. That's adenine, guanine, cytosine, thymine, etc. When these are bound to the phosphate backbone of DNA and RNA, the nitrogen bases are called as nucleotides. As you can see in the figure, have taken a simple model of double helix DNA structure where the coiling is nothing but the phosphate backbone, and in between the colored stripes are the ladder ties are nothing but these are the different kinds of nitrogenous bases which have formed the complementary bonding with the help of hydrogen bond in order to make it stable and to make it stable. So hope you understand the session, and thank you for watching. Thank you so much.